Hi, and thank you for tuning into the Nature Conversations podcast. I'm Dr Adam Forbes. I'm a forest ecologist who specialises in the restoration of New Zealand's forest ecosystems. This podcast isn't about me, however. The purpose of the Nature Conversations podcast is to capture knowledge and insights from remarkable people and to profile important issues for ecology and nature conservation. In this episode, I welcomed Dr Shaun Wilkinson onto the podcast. Shaun has a background in DNA research and has in recent times established a lab offering eDNA analytical services to anyone interested in collecting eDNA data. It was great to have Shaun on the show to hear all about his work with eDNA.
A lot of farmers who we are working with are really interested in figuring out, what is the current diversity of their stream, aside from just the stuff they can see, like the plants and the fish and the freshwater crayfish. There's a huge amount of biodiversity swimming around down there, and this is a great new lens to be able to look at that biodiversity and start figuring out which are the threatened species that are in their waterway, which are the invasive species, and what might they be able to do about that. I'm really starting to see some good impacts, and I get really excited about thwarting these invasions before they become a real problem, because if we can catch them while they're still small and eradicate them, that saves us a heck of a lot of money with some of these containment programs.
Well, it's my pleasure to welcome Shaun Wilkinson to Nature Conversations. Thank you very much for joining, Shaun. How are you today?
Very well, thanks, Ian. Thanks for having me on the show. Very excited to hear about this podcast and honoured to be one of the first episodes.
Look, I'm so excited to have you here and to hear about your insights and expertise in eDNA. Would you mind just starting by explaining a little bit about your professional background and how you came to work in the field of environmental DNA?
Yeah, sure. I actually started my career as a chef, and I was working as a head chef at a busy Palmerston North restaurant for around 3 years. And after, preparing one too many Caesar salads, I left the industry, worked in commercial kitchen setups for around 3 years, doing sales and that type of thing. And then I think it was my father-in-law who learnt about my maths grades, and he said I have to go back to university. So at the ripe old age of 27, I went back to Victoria University, did a Bachelor of Science in Marine Biology, did a honours PhD in coral genetics under Professor Simon Davey, postdoctoral research up in East Timor, and that's when I first learnt about environmental DNA. I was working in collaboration with the Trend Lab, Traits and Environmental DNA Lab over at Curtin University in Perth under Professor Mike Bunce, with Michael Statt there as well. I was taking DNA samples from the coral reefs of East Timor back over to Perth and analysing them. And that's when I really got the bug for eDNA. So once I got back from East Timor, there was a bit of a career lull where I was applying for postdoctoral research grants and not having much luck. Very few available in New Zealand. Bit of a leaky academic pipe, and I was one of the drips. So I kind of got out of academia at that point and decided to start my own commercial eDNA laboratory. It seemed like there was a big gap in the market here in New Zealand and abroad. The rest is history, really.
So the lab name's Wilderlab, is that right, Shaun?
Wilderlab, yeah. So it's kind of like a lab in the wilderness. Think of it like that.
I see. Oh, that's so cool. Thank you for explaining that. So can you describe to me, for a starting point, what is eDNA?
Yeah, so eDNA generally refers to the genetic traces of living things, the molecular kind of signals that they leave behind in their environment when they're swimming around or crawling around. And that kind of contributes to this big genetic juice in the rivers and oceans and things that keeps kind of accumulating. And it's generally tied up in molecules, so cells and gametes and that type of thing. But we can filter out those biological molecules reasonably easy, just using standard kind of filters and syringes, purify the DNA and run that through a high throughput DNA sequencing machine. That pretty much just converts the DNA into text files that we can then use, match it against a reference database of known gene sequences of known species, to provide a big list of the different species that are present in the environment. So it's a little bit like a crime scene investigation, in that you go to a crime scene and you swab for DNA and run it against the database to get a match. Here we're doing a similar sort of thing, but with species. So we're using a big species database that's all publicly available. It's called GenBank. Everyone who works on this species of moth or that species of tree will upload a genetic sequence to GenBank, and we can use that information to match these environmental DNA sequences against to provide that list of species.
So is it just water that this is relating to, or are there other mediums that you can collect samples from as well?
You can pretty much collect DNA from almost anything. Water is the main one that we focus on, just because it seems to be the most straightforward way to collect DNA. We've got big water problems in New Zealand, as I'm sure everyone's aware. And so there's a big demand out there to be able to test water for DNA signals. But there's a burgeoning field of air DNA research happening at the moment. So people plucking DNA out of thin air using filters and wind socks and all sorts of things. You can collect it from soil, leaf litter, feces, scat from predators and things. Pretty much anywhere there's life, there's DNA, and so you can figure out a way to purify that DNA and run it through the sequencer.
Very interesting. So tell me, with regard to the business that comes through your lab, what would be the main applications, and are there any kind of on the fringe that are of note and interesting?
Yeah, so we are very much firmly focused on freshwater in our lab currently, just because, like I said, this is where all the big focus is in New Zealand. Huge focus to try to clean up our rivers and wetlands and things. Lots of invasive species getting in. And so our kind of bread and butter, I guess you'd say, is freshwater, and we work a lot with regional councils, other unitary authorities, Department of Conservation, MFE, the EPA, consultants who work on behalf of government or doing sort of infrastructure projects, lots of farmers, forestry and that type of thing. And so that's kind of the core of our business, and we're finding there's a lot more demand coming in for marine eDNA sampling as well, particularly from offshore, and we get all sorts of different random things. Lots of poos coming in these days. People wanting to analyse what things have been eating, and that seems to be a really growing field as well. Things like stoats and pigs, which seem to be feeding on Hochstetter's frogs and Archie's frogs and things. So lots of fecal matter coming in, and we even started to get into the honey business as well now. So people wanting to analyse honey to see one, what plants the bees have been working with, and two, what pests might be infesting the beehives.
So I'd be really interested to get from you a scenario, mainly for the listeners to get an appreciation, a bit more depth about how eDNA might be useful to them. So let's take, say, a farmer in New Zealand that is interested to know, say, about the health or about the status of a waterway that runs through their land. So what could eDNA tell them, and then how would they go about the process of implementing it?
Yeah, sure. So, incidentally, we do a lot of work with farmers around New Zealand. It seems to be a very popular tool for people, especially those engaged with sort of waterway restoration and that type of thing, even at any point along the process where they're just sort of starting to think about it or well developed in a restoration program. And it's also become part of the New Zealand FAP plus accreditation scheme. So we're getting a lot of samples coming in from farmers who are getting this sort of sustainability accreditation, showing that they're testing their local waterways and sort of doing something about it. A lot of the farmers who we are working with are really interested in figuring out, what is the current diversity of their stream, aside from just the stuff they can see, like the plants and the fish and the freshwater crayfish. There's a huge amount of biodiversity swimming around down there. And this is a great new lens to be able to look at that biodiversity and start figuring out which are the threatened species that are in their waterway, which are the invasive species, and what might they be able to do about that. And any restoration that they're working on, this is a great tool to be able to track the progress of those restoration efforts. So, for example, if someone retires a bunch of land alongside the river, plants some native trees along there, fences out the cattle, people want to be able to see that is having a positive impact on the river. And so by doing regular eDNA testing, they can track quite finely how the ecological health of that waterway is tracking through time. And it can be especially powerful if, for example, farmers are working together as a sort of a catchment group or a stream care group, and starting to do this at a larger scale. And by setting some goals around freshwater restoration, these tools are available to say, hey, currently we're sitting at a 6.1 out of 10. We want to get to a 7 out of 10 by this time, in 5 years' time. So having those goals and those really finely calibrated measurements to be able to say, this is our progress, this is where we want to get to, can be quite a powerful motivator for people.
I can imagine. And so is it as simple as saying that that an eDNA test will tell you, what is it, all living things upstream within the waterway? What does the sample actually represent?
So it doesn't tell you everything that lives in the waterway, and that's something that may happen in the future. But the issue is, to be able to target everything, you need to be have a complete reference database. So you have to have every single species living there uploaded in the database alongside its genetic signature, and we're just nowhere near that yet. So there's been a lot of work going into reference barcoding species. Much of it has been for the purpose of improving environmental DNA tools, but there is just a long way to go. And so we have all of New Zealand's freshwater fish species in the reference database now. We have around 2/3 of the aquatic insects. But once you start getting down into those sort of lower levels of bacteria and phytoplankton and zooplankton and things, it's anybody's guess how much diversity is really out there. And we've only captured just a very small fraction of that in those reference databases so far.
Okay, that sounds fair enough. And you mentioned, in terms of the results, you mentioned some numbers. So what do you get back in results? Is there some index score, or how are the results expressed?
Yeah, so usually, a standard eDNA test will give you back a big list of species, and you sort of go, okay, well, that's great. We've got, this species of fish and that species of plant. But there's a real demand out there for people to be able to condense all that quite complex information down into simple, interpretable metrics that they can understand and they can act on. So we spent a lot of time and a lot of effort developing, it's called a TICI, a Taxon Independent Community Index. And that effectively distills all that very complicated DNA sequence data down to a single number where people can use to say, this is the health of my waterway. And it's a little bit like the existing tools that are around, where you scoop up some bugs, and some bugs get high scores and some bugs get low scores because some are adapted to high quality environments and others are adapted to degraded conditions. You take the average of the scores and that's your index. This does the same thing but with sequence data. So it uses a machine learning approach and looks for DNA sequences that are associated with high quality habitats and others that are associated with degraded habitats. And it effectively finds a composite number, the average of all of those different sequences that are found in the sample, and that's your TICI index. And that works really, really well because it enables us to use all of the different data right from across the tree of life, bring in plants, bring in insects, bring in fish, bring in microbes and everything to build this index. And you get a really powerful number that is very robust, doesn't vary a lot between sample replicates, and can be used at quite fine scales to address progress or degradation.
That's fascinating. What a great tool. So would you mind just stepping me through, so let's take the farmer scenario again on their land. What's the process that person would go through from woe to go, from actually needing to I guess, get their sampling equipment from you, and then right through to what they'll receive at the very end?
Yeah, so we're all about making these tools accessible. That's the big mission for us, is to take the eDNA technology out of the laboratory and into the world so that people can use it and engage with it. And a big part of that has been creating these products and a user interface where people can jump onto the website, order a DNA sampling kit with their credit card, just like they would a human genomics kit to figure out what their ancestry is. Get the kit the next day and take it down to their local stream, take a sample. Very easy. It's about a 5-minute process. You get a pair of gloves and a filter and a syringe. You filter the water through the filter, discarding all the outlet water, and the preservative syringe that comes in the pack gets injected into the filter, and that's what keeps it fresh at room temperature while it's in transit. That then gets sent back to our lab and generally takes us about a week to process that sample, create a big list of species, put the Tiki index together, and send it out as an eDNA sample report. And that goes out as a spreadsheet. And we also have a nice interactive web report as well that people can access via the map application on our website. And that's got some nice pictures of all the different species that are in there. It's got the links through to the Wikipedia pages for the different species. And it's just all about engaging people and making it a more of a fun kind of exercise and getting people learning and thinking about what's in their waterway.
Could you give us an idea of indicative cost? Because I think some people will probably be listening to this going, that sounds great, but how much might I be spending? Would you mind just giving us a rough idea?
$290 plus GST per sample.
Great. Oh, that's really cool. Tell me, I'm interested, given your involvement in this technology and your background and your expertise, are there any avenues of this technology that you would in particular like to see developed in the future, or applications, or are there any directions that you'd like to see this move?
Definitely. I think we are, in New Zealand, one of the more proactive eDNA using countries. We've processed over 50,000 eDNA samples from around New Zealand to date, which is, per capita, I think would be one of the highest in the world. And so it's been really good to see, New Zealand Inc. come on board with this technology and really start to use it in anger. However, I still think we're really only scratching the surface of what it's capable of. Like I mentioned, we're mainly doing freshwater testing at the moment. Huge opportunities to roll this out into the marine environment to test for invasive marine species or the impact of aquaculture farms or offshore wind farms and that sort of thing. The eDNA, I think, is a real useful tool for particularly surveillance of pest mammals and things as we work towards predator-free 2050. We're going to need some really good sensitive tools for picking up surviving populations after some eradications. And once you start thinking about the field of human health, the applications are really just endless. So we've been doing COVID wastewater variant detection using this technology. You might remember from a couple of years ago when you were hearing about this proportion of Omicron and this proportion of Delta in the waterways. That was us working with ESR to roll that out. And that was just one little human health application. You can imagine with bird flu kind of on the horizon, we should be out there sampling ponds and things to capture the fecal matter of ducks and things as they're flying around, potentially spreading those kinds of viruses. Wastewater treatment plants, we could be picking up all the different flus and the different, other viruses that are in the population, tracking them to relatively fine spatial levels. And so the applications are really endless, and I really feel like we're just scratching the surface at the moment with the freshwater work that we're doing. The work that we're doing with communities around New Zealand, and this is largely driven by the Environmental Protection Authority through their program Waituwhera o Te Taio, which has been just a marvellous way to get eDNA kits out into the hands of those people that are on the ground in the regions doing the hard mahi to clean up their waterways and protect their waterways from invasive species. And that program has really just opened people's eyes to what this technology is capable of and how it can be used as part of restoration and environmental improvement. thousands of people have had opportunities to take eDNA samples from school children all the way through to, experienced practitioners through this effort. And this has been really quite a learning experience for us as well in terms of, some of the cultural considerations about the use of DNA and monitoring in New Zealand. Not everyone is as excited as me to post the DNA sequences online for everyone to see. There's a fair bit to work through, I think, there, with particularly with Indigenous communities and their desire and rights to hold their data and own their data. So it's been quite a thorough learning experience for us as well. And one of the big outcomes that's come out of it for us is that we now, when we do eDNA data for people, all of the rights to that data is signed over to the person who does the testing, who purchases the DNA kit. We don't hold any right over any of the data, which is kind of a progressive thing, I think, for a sequencing lab to do. And it just means that people have agency over their own data. They can choose to make it public if they want to, or they can choose to keep it private and just share it amongst their friends.
So, Shaun, are you starting to see some positive impacts from eDNA monitoring in particular, rather than just other types of monitoring that people might be doing?
Yeah, now that we've been going sort of 5 years and have started to build up quite a rich data set, there are definitely some examples starting to come out of where we're seeing some real tangible environmental positive outcomes from the use of eDNA tools. There's been some really good examples down south, working with the Department of Conservation, looking for threatened native non-migratory fish, the Galaxis fish. There's one in particular, the Clutha flathead Galaxius, which is a critically endangered species confined to just a few hectares of habitat. And it's a really tricky one to try to find using traditional survey techniques. People will try electrofishing for them, but their net has its own invasive kind of problems. Plus, they're very cryptic, and you have to sort of monitor very long stretches of waterways to try to find them. And so these operations staff at the Department of Conservation have been able to find new populations of these critically endangered Galaxis, and then they've been working to install trout barriers as well. So they're excluding the trout from this habitat, and they're already starting to see big improvements, lots of juveniles, the adults starting to move down as far as the trout barriers, and so those populations are really starting to rebound. On the invasive species front as well, we've got some really good examples, particularly coming out of the Manawatu and the Hawke's Bay, where eDNA monitoring has been used, sort of regular eDNA routine monitoring, mainly for the purposes of sort of fish detections, are starting to pick up these invasive plants that are starting to spread through the region. And there was one recently in Lake Whatuma in the Hawke's Bay, an outbreak of alligator weed, which is definitely not supposed to be in the Hawke's Bay region. That was picked up using a routine eDNA test by Hawke's Bay Regional Council, and they've now diverted about $100,000 of budget to eradicate that and just get rid of it. And wouldn't have known about it had they not been doing eDNA testing. And there's a few of those examples starting to come through now. So I'm really starting to see some good impacts, and I get really excited about thwarting these invasions before they become a real problem, because if we can catch them while they're still small and eradicate them, that saves us a heck of a lot of money with some of these containment programs.
A stitch in time saves nine, doesn't it, Shaun? That's really good. I can see the point in what you're saying there, just to be on the front foot and pick things up before they take off. That's huge merit in that. And that Galaxid story is excellent too, just to be able to detect a cryptic species. It's superb.
Yeah, there's also another example where we can actually use eDNA, really front-footing it this time, for species discovery. So there's a population of, it's called a gravel maggot. The genus name is Smeagol. It's a little slug that lives in the sand, between the sand grains on a beach in Wellington. And it's again critically endangered, confined to just a few hundred square meters of habitat. And we were able to picked that up in Wellington, which is great. And then all of a sudden we had a hit on the same or very similar genetic signature from the west coast of the South Island, in a very remote marine reserve, which was very exciting because it looks like there's another population or possibly another species of these gravel maggots inhabiting the sand grains down on the west coast of the South Island.
So what sort of sample was collected to detect the gravel maggot? Was it a sample of water, or how was that done?
Yeah, it was just a seawater sample that a dock ranger had taken for the purposes of marine reserve monitoring. So just to get a bit of an idea of the biodiversity inhabiting this remote marine reserve. And I think he was just as excited as we were to pick up that cryptic species.
Yeah, and how else would it be done? That's amazing.
Yeah, so that's it in a nutshell.
Very good. Well, thank you very much for joining me on the podcast, Shaun. Really good to hear about your work, and I wish you all the best with it.
Thanks very much, Adam. Appreciate it.
That was a fascinating discussion with Dr. Shaun Wilkinson. His passion for eDNA is clear, and it's wonderful to see he has found an avenue to keep contributing to this part of science. Environmental monitoring can be subject to financial and practical barriers, and eDNA is looking to be one way of navigating around some of those barriers. It is great to know that Shaun will be continuing his work in this field, and it will be very interesting to see how the eDNA technology develops in the future and where in particular Shaun continues to develop the monitoring services on offer. Thanks very much, Shaun, for your contribution to the Nature Conversations podcast, and I wish you all the best with your ongoing work with eDNA. If you like this episode, be sure to subscribe so you can receive future episodes. If you'd like to help with the production of this podcast, you can do so via my Patreon link in the episode description. Thanks very much for joining, and I look forward to sharing another episode with you soon. Bye for now.